Mycobacterium is a genus of actinobacteria, given its own family, the Mycobacteriaceae. The genus includes pathogens known to cause serious diseases in mammals, including tuberculosis and leprosy. The Greek prefix myco means fungus, alluding to the way mycobacteria have been observed to grow in a mold-like fashion on the surface of liquids when cultured. Microbiologic characteristics. Mycobacteria are aerobic and non-motile bacteria that are characteristically acid-alcohol fast. Mycobacteria do not contain endospores or capsules and are usually considered gram-positive. While mycobacteria do not seem to fit the gram-positive category from an empirical standpoint, they are classified as an acid-fast gram-positive bacterium due to their lack of an outer cell membrane. Mycobacterium marinum and perhaps M. bovis have been shown to sporulate. However, this has been contested by further research. The distinguishing characteristic of all Mycobacterium species is that the cell wall is thicker than in many other bacteria, which is hydrophobic, waxy, and rich in mycolic acids mycolates. The cell wall consists of the hydrophobic mycolate layer and a peptidoglycan layer held together by a polysaccharide, arabinogalactan. The cell wall makes a substantial contribution to the hardiness of this genus. The biosynthetic pathways of cell wall components are potential targets for new drugs for tuberculosis. Many Mycobacterium species adapt readily to growth on very simple substrates, using ammonia or amino acids as nitrogen sources and glycerol as a carbon source in the presence of mineral salts. Optimum growth temperatures vary widely according to the species and range from 25 °C to over 50 °C. Some species can be very difficult to culture, sometimes taking over 2 years to develop in culture. Further, some species also have extremely long reproductive cycles. M. lepi may take more than 20 days to proceed through one division cycle, making laboratory culture a slow process. In addition, the availability of genetic manipulation techniques still lags far behind that of other bacterial species. A natural division occurs between slow larvae and rapid larvae growing species. Mycobacteria that form colonies clearly visible to the naked eye within 7 days on subculture are termed rapid growers, while those requiring longer periods are termed slow growers. Mycobacteria cells are straight or slightly curved rods between 0.2 and 0.6 aa micrometer wide by 1.0 and 10 aa micrometer long. Pigmentation. Some mycobacteria produce carotenoid pigments without light. Others require photoactivation for pigment production. Photochromogens produce non-pigmented colonies when grown in the dark and pigmented colonies only after exposure to light and reincubation. M. consaza, M. marinum, M. simeri. Scotochromogens produce deep yellow to orange colonies when grown in the presence of either the light or the dark. M. scrofulaceum, M. gordoni, M. xenopi, M. sulgi. Non-chromogens, non-pigmented in the light and dark or have only a pale yellow, buff or tan pigment that does not intensify after light exposure. M. tuberculosis, M. avium intracellular, M. bovis, M. ulcerans, M. fortuitum, M. cheloni. Staining characteristics. Mycobacteria are classical acid-fast organisms. Stains used in evaluation of tissue specimens or microbiological specimens include Feit's stain, Ziehl-Neelsen stain, and Kinune stain. Mycobacteria appear phenotypically most closely related to members of Nocardia, Rhodococcus and Corynebacterium. Ecological characteristics. Mycobacteria are widespread organisms, typically living in water and food sources. Some, however, including the tuberculosis and the leprosy organisms, appear to be obligate parasites and are not found as free-living members of the genus. Pathogenicity. Mycobacteria can colonize their hosts without the hosts showing any adverse signs. For example, billions of people around the world have asymptomatic infections of M. tuberculosis. Mycobacterial infections are notoriously difficult to treat. The organisms are hardy due to their cell wall, which is neither truly gram-negative nor positive. In addition, They are naturally resistant to a number of antibiotics that disrupt cell wall biosynthesis, such as penicillin. Due to their unique cell wall, they can survive long exposure to acids, alkalis, detergents, oxidative bursts, lysis by complement, and many antibiotics. Most mycobacteria are susceptible to the antibiotics clarithromycin and rifamycin, but antibiotic-resistant strains have emerged. As with other bacterial pathogens, surface and secreted proteins of M. tuberculosis contribute significantly to the virulence of this organism. There is an increasing list of extracytoplasmic proteins proven to have a function in the virulence of M. tuberculosis. Medical classification: Mycobacteria can be classified into several major groups for purpose of diagnosis and treatment: M. tuberculosis complex, which can cause tuberculosis, M. tuberculosis, M. bovis, M. africanum, and M. microti. M. lepi, which causes Hansen's disease or leprosy. Non-tuberculous mycobacteria are all the other mycobacteria, which can cause pulmonary disease resembling tuberculosis, lymphadenitis, skin disease, or disseminated disease. Mycosides: Mycosides are phenolic alcohols that were shown to be components of Mycobacterium glycolipids that are termed glycosides of phenylthioceryl dimycosauricate. There are 18 and 20 carbon atoms in mycosides A and B, respectively. Genomics: Comparative analyses of mycobacterial genomes have identified several conserved indols and signature proteins that are uniquely found in all sequenced species from the genus Mycobacterium. Additionally, 14 proteins are found only in the species from the genera Mycobacterium and Nocardia, suggesting that these two genera are closely related. Species: Phenotypic tests can be used to identify and distinguish different Mycobacteria species and strains. In older systems, Mycobacteria are grouped based upon their appearance and rate of growth. However, these are simply zoomorphies, and more recent classification is based upon cladistics. Slowly growing, Mycobacterium tuberculosis complex. Mycobacterium tuberculosis complex members are causative agents of human and animal tuberculosis. Species in this complex include M. tuberculosis, the major cause of human tuberculosis, M. bovis BCG, M. africanum, M. conneti, M. capi, M. microti, M. pinophytii. Mycobacterium avium complex. Mycobacterium avium complex is a group of species that, in a disseminated infection but not lung infection, used to be a significant cause of death in AIDS patients. Species in this complex include M. avium paratuberculosis, which has been implicated in Crohn's disease in humans and is the causative agent of Johns disease in cattle and sheep, M. avium sylvaticum, M. avium homonissuis, M. columbiense, M. indicus pruni. Mycobacterium gordoni clade, M. isiaticum, M. gordoni, Mycobacterium consaza clade, Mg, M. consaza, Mycobacterium non-tromogenicum terrae clade, M. hibernau, M. non-tromogenicum, M. terrae, M. trivial, mycolactone-producing mycobacteria, M. ulcerans, which causes the Buruli, or Bairnsdale, ulcer, M. pseudoschottzia, M. schottzia, Mycobacterium simeri clade, M. triplex, M. genavans, M. florentinum, M. lentiflavum, M. pallister, M. cubici, M. parascrophulaceum, M. heidelbergens, M. interjectum, M. simeri, ungrouped, M. branderi, M. cooker, M. slatum, M. bumicum, M. homophilum, M. mulmans, M. sulgi, M. lepi, which causes leprosy, M. lepraemurium, M. lepromatosis, another cause of leprosy, described in 2008, M. botanians. M. chimera, M. conspicuum, M. doricum, M. fascinogenes, M. hercessionines, M. intracellular, M. lacus, M. marinum, M. monaciens, M. montiferens, M. morale, M. nebraskans, M. saskatchewanense, M. scrophulaceum, M. chiamoidae, M. tusheri, M. xenopi, M. yonganines, intermediate growth rate, M. intermedium, rapidly growing, Mycobacterium chelonie clade, M. abscissus, M. chelonie, M. bollichia, Mycobacterium fortuitum clade, M. fortuitum, M. fortuitum subsp. Astamidoliticum, M. boenicum, M. peregrinum, M. porcinum, M. senegalens, M. seticum, M. nebuliensans, M. haustonans, M. mucogenicum, M. megaritans, M. brisbanans, M. cosmeticum, Mycobacterium parafortuitum clade, M. parafortuitum, M. austroafricanum, M. dienhoferi, M. hoteleri, M. neorum, M. fredericksborgens, Mycobacterium vaccae clade, M. aurum, M. vaccae, cf. M. keti, M. phallax, ungrouped, M. confluentis, M. flavescens, M. madagascarians, M. flu, M. smegmatis, M. goodyeari, M. wielinski, M. thermorecistibule, M. gadium, M. comosens, M. obuents, M. sphafni, M. agri, M. acians, M. alvu, M. aupans, M. bruni, M. canarisens, M. tubuents, M. conceptionines, M. darvala, M. elephantis, M. gilvum, M. hosaicum, M. holsaticum, M. immunogenum, M. macilians, M. moriograns, M. cycrotolerans, M. pinivorans, M. van Baarlnii, M. pulveris, ungrouped, M. erosians, M. aubignines, M. capi, M. chlorophenalicum, M. fluoroanthonivorans, M. kumamotonants, M. novocastrans, M. palmants, M. ficacum, M. poriferae, M. rhodioschii, M. solens, M. tocaeans, Mycobacteriophage. Mycobacteria can be infected by Mycobacteriophage, bacterial viruses that may be used in the future to treat tuberculosis and related diseases by phage therapy. References. Further reading. External links: Tuberculist, Genome Annotation Database, MTB Sysborg, Genome Annotation Database from the Institute of Genomics and Integrative Biology, TB Structural Genomics Consortium, Structures of Mycobacterium Tuberculosis Proteins, MYCDB, Mycobacterium Database, TBDB, Tuberculosis Database, Mycobacterium Genomes and Related Information at Patrick, a bioinformatics resource center funded by NIAID, frequently asked questions about NTM lung disease, precise identification of mycobacteria, glycolic acids mycolates. The cell wall consists of the hydrophobic mycolate layer and a peptidoglycan layer held together by a polysaccharide, arabinogalactan. The cell wall makes a substantial contribution to the hardiness of this genus. The biosynthetic pathways of Sowei mycobacteria have been observed to grow in a mold-like fashion on the surface of liquids when cultured. Microbiologic characteristics. Mycobacteria are aerobic and non-motile bacteria that are characteristically acid-alcohol fast. Mycobacteria do not contain marinum and perhaps M. bovis have been shown to sporulate. However, this has been contested by further research. The distinguishing characteristic of all Mycobacterium species is that the cell wall is thicker than in many other bacteria, which is hydrophobic, waxy, and rich in endospores or capsules and are usually considered gram-positive. While Mycobacteria do not seem to fit the gram-positive category from an empirical standpoint, they are classified as an acid-fast gram-positive bacterium due to their lack of an outer cell membrane. Mycobacterium is a genus of actinobacteria, given its own family, the Mycobacteriaceae. The genus includes pathogens known to cause serious diseases in mammals, including tuberculosis and leprosy. The Greek prefix myco means fungus, alluding to the